So far, we've looked at primary structure, which is just the amide bonds that link amino acids together in this linear chain. But what happens is when the primary sequence of the polypeptide, so primary structure, when it folds, so it can fold into regularly repeating structures, we get what's called secondary structure. So secondary structure is a result of the folding of the primary sequence of the polypeptide. Secondary structure results from hydrogen bonding. So we're talking about hydrogen bonding between the amide hydrogens and the carbonyl oxygens of the peptide bonds. Now, if I just isolate a little part of a protein or a peptide, right, we're going to have, we have our alpha carbon here and we have our R group, but we have a carbonyl, and carbonyls can participate in hydrogen bonding. And then we have the amide hydrogen, and of course that can participate in hydrogen bonding as well. But what we're going to see is that not every region of a protein is going to have a clearly defined secondary structure. Some are going to be random and some will be non-regular. The most common type of secondary structure is what we call the alpha helix, and it's a right-handed helix, just like an ordinary screw. Anyhow, it's coiled and it's helical, of course. So the important features are that in that secondary structure, or that alpha helix, each amide hydrogen and each carbonyl oxygen is going to be involved in hydrogen bonding. And that's what's going to lock that helix into place. So we're going to see hydrogen bonds. They're going to lock that coil together, if you will. So the hydrogen bonds are parallel. They're going to be parallel to the long axis. So here's the long axis of the helix, right? It runs up and down like this. And the hydrogen bonds are going to be parallel to it. Again, the helix is right-handed, and then we have a repeat distance or pitch, which is about 5.4 angstroms, and we have roughly 3.6 amino acids per turn. So here's a representation of the alpha helix. So you can see the right-handed helix going this way, right, and it's turning around and coming back like this, and then you can see all the bonds. So for example, here's a carbonyl and here's a nitrogen and so on and so forth. But what you can see is what holds that helix together is that there's a hydrogen bond between, if we look at this carbonyl here, you can see there's a hydrogen bond between it, one of the lone pairs on that oxygen, and this hydrogen, which is from an amide bond, of course. Right, if we look at another one here, you can see this is another nitrogen, so the nitrogens are blue, and there's a hydrogen bond with an oxygen on a carbonyl there. So that's what's holding this alpha helix together. If you were to look down that alpha helix, if you were to look straight down like that, what you would see is what's shown over here. So what's interesting is that all the R groups, so what identifies all these different amino acid residues, those R groups are all pointing outward. Alpha helices can form something that we call fibrils, and fibrous proteins are arranged in a secondary structure of fibers or sheets with only one type of secondary structure, so the alpha helix, and it has repeated coiling of the helices. So here's an example of a hair fiber. So a hair fiber is made from what we call an alpha keratin, and so that's just made from a bunch of alpha helices put together, and you don't have to know all these different things like a protofibril and a macrofibril, but the idea is that when we take an alpha helix and we put three of them together, we get what's called a protofibril, and there's going to be extended hydrogen bonding here, which leads to a lot of strength, and then we put them together into something even larger called a macrofibril. So the proteins of hair consist almost exclusively of polypeptide chains coiled up into alpha helices. A single alpha helix is coiled in a bundle with two other helices to give the three-stranded superstructure called a protofibril that is part of an array that we call a microfibril. The major structural property of a coiled coil superstructure of the alpha helices is its great mechanical strength. After the alpha helix, the second most common type of secondary structure is what we call the beta-pleated sheet. And the beta-pleated sheet appears similar to the folds that you would see in fabric like draperies, or maybe the corrugation in corrugated cardboard. All of the carbonyl oxygens and amide hydrogens are involved in hydrogen bonds with the chain nearly completely extended. So what that means is if you were to take a peptide or a protein and you were to write out the primary structure and you were to put it all out in a line, what's going to happen is that if we have our N-terminus here, let's say, and our C-terminus here on a protein, and then if we have the N-terminus and the C-terminus, well, the C-terminus of another protein, what's going to happen is that we can have hydrogen bonding between the carbonyls on one of these peptide chains or protein chains, and between the nitrogen hydrogens on the other chain. So there's going to be hydrogen bonding like this. And what happens is when the protein chain folds like this so that the N-termini, so when they line up like this, I call that a parallel beta pleated sheet. If I have my N-terminus here, and then of course I would say, okay, well here's a carbon here, and then if I were to flip it over like this, so I have a nitrogen here, and then my carboxylate is down here, well, this is what we call an antiparallel. But again, the antiparallel beta pleated sheet is going to be held together from hydrogen bonds from the carbonyl oxygens and the hydrogens that are on the amide nitrogens. So to review, the two types of secondary structure are the alpha helix and the beta pleated sheet.